Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about the protein folding and molecular chaperone, the mechanism of protein folding and molecular chaperone, that how the molecular chaperone are involving during protein folding. Okay, so in the previous video lecture, if you guys remember, we just talked about the molecular chaperone overviews, and we talked about the molecular chaperone function, and we talked the types of the molecular chaperone, and we also talked a little bit protein folding pathway, as we can see, and we talked about the protein folding, a little bit protein folding, that how protein will be folded, and we just talked about their introduction as well. If you missed this video lecture, it is present into my channel. You can go back and watch, or don't worry, inshallah I will give the link of this video in the description box, then you will check it. Okay, so let me erase this one portion and let me start the mechanism of protein folding in molecular chaperone. Okay guys, so let me write mechanism. Now what we have during translation, the protein folding is sometime a co-translational process, or sometime it will be occur after the translation. We also say like that, but most of the time protein folding will be occur after the translation. Now what happened, this is, let's suppose, the linear structure of the protein. As you can see, they have the N terminus and C terminus. Now what happened in the first stage, first of all, two types of the molecular chaperone are involved. In the first we have the DNA J. We call it DNA J in prokaryotes, while in eukaryotes we have the HSP40. So this is the first protein which are involved. Now after that, the second molecular chaperone we call it DNA K with ATP, or in prokaryote or in eukaryotes we call that HSP70. So HSP70, it is also the ATP. It have also this ATP. So what happened with the addition of these two protein or molecular chaperone, this linear form of the protein will be converted into a non-linear or into a secondary structure of the protein. So this is the primary structure, so it will be converted to a secondary structure, as you can see. So this is the second, this is the N terminus and this is the C terminus. So this is the secondary structure. We also call it tertiary or quaternary structure of the protein, because tertiary or quaternary structure of the protein are the functional. Okay. Now this is also the non-linear structure of the protein, and this is also called as native protein. Now this is the nascent protein. So what happened after that, this folding form of the protein required energy. So the energy will be obtained by this ATP. So this ATP will be converted into ADP plus pi, inorganic phosphate. So two phosphate are removed, as you can see, which we call it inorganic phosphate, and it is used as a energy. So then energy will be required, due to which the protein will be in a folded form, as you can see. Now this protein is in a functional form. This protein perform their function easily. But sometime what happen, these two protein or these two molecular chaperone are failed to perform their function, and somehow we can get the secondary structure of the protein, or tertiary or quaternary, whatever you can say. We get the folded form of the protein, but this folded form of the protein still, this folded form of the protein still don't have any function. So this folded form of the protein will sometime further modify. It will be converted into a tertiary or quaternary structure. Let's suppose this is the secondary structure of the protein, which is non-functional, let's suppose. Or in some cases it will be a functional. In most of the cases it is functional, but in a rare cases it is non-functional. So what happened then, after that a next molecular chaperone will involved, and we call that GROW EL and GROW ES. In prokaryotes we call that grow EL and grow ES, while in eukaryotes we have the protein HSP10 and HSP60. So these are two other protein, and these two protein are also actually the ATP. Okay, so these two protein are involved to convert this folded form of the protein or convert this secondary form of the protein into a tertiary structure, and somehow we will get the protein structure like that. So this is the tertiary structure of the protein. Now this quaternary or tertiary structure of the protein are responsible for the function. This protein will not more further are converted into folded form, and during this way this ATP will also give energy, and it will be converted into ADP plus inorganic phosphate, and the energy will required by this protein to make a tertiary structure of the protein, as you can see. So all this folding process will be performed during at a temperature of what we can say 30 to 42 centigrade, and we also call that heat shock protein, as you can see, because this molecular chaperone can perform their function at that temperature. So this is all about the protein folding phenomena and the function of molecular chaperone during protein folding, that how it will perform their function. And this is actually the protein folding phenomena in both prokaryotes and in eukaryotes, but the name are different, as you can watched. So this is all about the protein folding mechanism. I hope you get idea about that, but if still you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and definitely subscribe the channel to get more interesting videos like that. Thank you so much for watching.